Hello, welcome to Jera Board. In today's episode, we're going to be discussing why cancer is very difficult to cure. We have harnessed electricity. We have sequenced the human genome. We have virus, antibiotics, antifungi, and antiviral drugs to diseases that were incurable. And last, we have eradicated smallpox. But billions of dollars have been spent on the research of cancer. They haven't found a solution for disease that affects more than 15 million people worldwide and their families inclusive. Cancer arises as normal cells accumulate mutations. Most of the times, cells detect mutations or DNA damage and either fix them or destroy them. However, some mutations allow cancerous cells to grow unchecked and invade nearby tissues or even metastasize to distant organs. Cancers become almost incurable once they are metastasized. And cancer is incredibly complex. It's not just one disease. It has over 100 different. I don't have a magic bullet that can cure all of them. For most cancer treatments usually include a combination of surgery to remove tumors and radiation with chemotherapy to kill any cancerous cells left behind. Hormone therapies, immunotherapy, and targeted treatments tailored for a specific type of cancer are sometimes used too, and the patient becomes cancer-free. But they are far from 100% effective, 100% of the time. We are beginning to understand a few types of the problems scientists have. We need new, better ways of studying cancer. Most cancer treatments are developed using cell lines given in labs from cultures of human tumors. These culture cells has given us certain insights about cancer genetics and biology, but they lack much of the complexity of the tumor in an actual living organism. It's frequently the case that new drugs which work on these lab-grown cells will fail in clinical tests on the real patients. One of the complexities of aggressive tumors is that they have multiple populations of slightly different cancerous cells. Over time, distinct genetic mutations accumulate in cells in different parts of the tumor, giving rise to subclones. For example, aggressive brain tumors called glioblastomas, which have as many as 6 different types in a single patient. And it makes treatment different because a drug that works on one patient will not work on another subclone. Here is another challenge. A tumor is a dynamic, interconnected ecosystem where cancer cells constantly communicate with each other and with healthy tissues nearby. They can induce normal cells to become blood vessels that fill the tumor and remove waste products. They can also interact with the immune system to actually suppress its function, keeping it from recognizing and destroying cancer. If we could learn how to shut down these lines of communication, we will have a better shot of vanquishing the tumor forever. Additionally, mounting evidence suggests that we need to figure out how to eradicate cancer stem cells. In a theory, even if the rest of a tumor shrinks beyond detection during treatment, a single growth of a new tumor, figuring out how to target these stubborn cells might help prevent cancers from coming back. Even when we solve these problems, we might face new ones because cancer cells are masters of adaptation. Even with all we don't know as humans, the average mortality rates of most kinds of cancer have dropped drastically since 1970. Scientists are learning every day, and as new piece of information gives us more tools to our arsenal. Please stay safe